Okay. Hereby I open this academic ceremony in which Miss Melissa Schepers will defend her academic thesis, PDE4 Gene Inhibition, a Novel Approach to Treat Myelinating Disorders. May I invite you to present a summary of your studies and the conclusions of your thesis in the next 15 minutes. I give you the word.
Thank you, Prorector. Dear Corona, dear family, friends, and colleagues, it's a real pleasure for me to finally stand here in front of you today and tell you a bit more about what I've been doing over these past years during my PhD research. My research is entitled PDE4 Gene Inhibition, a Novel Approach to Treat Demyelinating Disorders, and in the upcoming 15 minutes, I will summarize my main findings. But first, let us start with a journey through the body. Our body is like a complex machine, relying on a control center for proper functioning. And in our body, this control center is called the nervous system. The nervous system can be subdivided into two main parts. On the one hand, we have the brain and spinal cord, which together make up the central nervous system. On the other hand, we have nerves running from our central nervous system to the rest of our bodies, such as the limbs and extremities. And this is called the peripheral nervous system. Now, for our body to be optimal functioning, this peripheral and central nervous system needs to be in continuous communication with each other. Now, in our journey through the body, we now encounter a specialized cell type, which we called neurons. And these neurons are the typical nerve cells, which make up a nervous system and are responsible for that communication within and between these nervous systems. Now, if we would imagine just a normal neuron without any special features, and a signal would arrive in that neuron, it needs to travel over the neuron to the next one. But this signal would encounter some challenges. First of all, its travel would take a rather long time, and it would also decrease in intensity. Luckily, nature has provided a solution for this, because in the proximity of these neurons, another specialized cell type is there, which we called oligodendrocytes. And these oligodendrocytes, they make a fatty substance that we call myelin. And that myelin is an electrical insulator, similar to the isolation around an electrical wire. If now a signal would arrive, its travel would be much faster, and it would not decrease in intensity. And this then leads to proper functioning of our muscle movements, memory, attention, and sensory. But what if something fundamentally goes wrong in this organization? An example of what can go wrong is the disease multiple sclerosis, or shortened MS. Because in MS, the immune system turns on you. Normally, we're constantly exposed to possible invaders, such as bacteria or viruses, but our immune system is there to prevent damage. However, in MS, the gateway to the central nervous system is open, and the immune system becomes overactive. Once this immune system, which is overactive, is then in the central nervous system, it mistakenly recognizes the myelin as a foreign substance, and it launches an attack on it. This leads to the loss of myelin, a process we call demyelination. And this then also has a lot of impairments accompanied with it. As such, MS patients experience problems with movement and vision loss. But luckily, our body is then again capable of coping with this to a certain extent. Because what is also in our brain and spinal cord are a cell type which we call OPCs. And these OPCs are the baby oligodendrocytes. They're there and they have the capacity to become an oligodendrocyte. So when there is this damage, these OPCs will become new oligodendrocytes, and they will remake myelin, a process we call remyelination. And this then leads to a certain level of restoration of our body functions. However, for an unknown reason in MS, this process eventually fails, leading to a buildup of symptoms. So if we want to help MS patients, what can we do? We can either temper the immune system so that no further damage would happen, or we can assist the repair cells so that more of that remyelination, that repair process will happen. How can we do that? For this, we need to take a closer look at what is happening within a cell. Because within a cell, we have this good substance, the substance we call cyclic AMP. And that cyclic AMP, it's good. It's there to keep your immune cells in a resting state, and it is important for cells to mature. However, what's also in the cell is a Pac-Man-like protein, which we call PDEs, and they chase and break down this cyclic AMP so that you have less of this good substance in your cell. So if we could halt this action of these Pac-Mans, that would be ideal, because then we can keep the cells in a resting stage, or we can cause maturation of these cells. So said, so done. Lessons learned from the past. Indeed, there was already a so-called PDE4 inhibitor that was tested, and this halted the action of all the Pac-Mans, so that they could no longer break down that cyclic AMP. And indeed, it was seen that that general Pac-Man inhibitor was beneficial because it reduced the overactivation of the immune system, and it caused repair to happen. However, because these Pac-Mans are everywhere in our body, stopping all these Pac-Mans lead to side effects, such as nausea and vomiting. So this inhibitor cannot be used for treating patients. Therefore, we took a closer look at these Pac-Mans and what is happening in the cells, and we could see that there are different colors of these Pac-Mans, different forms that are pretty much similar. And these we call PDE subtypes. They all belong to that Pac-Man family, but there are subtle differences between them. So we were wondering, can we use these differences in our advantage? And in science, we always work around one central question. We call this a research question. And that research question is something that we aim to answer with our experiments. And my research question during my PhD was, can we halt these PDE4 subtypes, these distinct colors, to treat MS? As a general approach, I made use of both mouse and human-based experiments. So let us first look at my mouse results. Now, what's important is that a mouse is not just a tiny human. There are big differences between them. As such, for example, a mouse does not spontaneously develop MS. So we need to use a model, something that mimics a bit the disease of MS. Now then, there's another problem. There's not one perfect model that mimics exactly MS. Therefore, in my study, I made use of two models. First is an inflammation-based model. The other one allowed me more to look at repair. In my inflammation model, a lot of these bad, overactive immune cells are present, but not that much of demyelination, the loss of myelin, is happening. In my other model, the repair model, there's not that much of the overactive immune system, but there's way more of that demyelination happening. So using both models, it allowed me to investigate both key hallmarks of MS, inflammation and demyelination. In my inflammation model, I either not treated the animals, just to look at the effect of the disease on the mouse itself, or I treated the mice with a general PDE4 Pac-Man inhibitor, the one that was good but had these side effects, or I used two specific inhibitors, targeting either only the purple Pac-Mans or the blue Pac-Mans. Now, because there's so much inflammation and overactive immune system present in these animals, this actually is showed by a level of paralysis. These animals experience paralysis. So if we do not treat our animals, what we can see over time is that there's an increase in this level of paralysis in these animals. But when we then treated the animals with our general PDE4 Pac-Man inhibitor, we could see that the level of paralysis was dramatically lowered, as expected, because it was known that it also worked already. If we then used our specific inhibitors, we could now see that targeting the blue Pac-Mans did nothing. These animals still had a high level of paralysis, but now the purple PD4B subtype Pac-Man, if we stopped that action, we could see that the paralysis level was very much lower, that this really was similar to what we saw with the general inhibitor. And this means that what we saw when we stopped all the Pac-Mans, all the PDE4s, was due to the halting of the action of the purple PD4Bs. And if we then looked at the central nervous system of these animals, we could indeed see that there was this shift in immune cells, that there were way more of these good immune cells and less of these bad overactive ones. Now, if I then looked at the repair model, the one that allowed me to look more at the de- and remyelination aspect of MS, I wondered, the blue one is not doing anything in that inflammation model. Could it have a potential here in this repair model? But now, because there's not that much of immune system activation, I could not evaluate paralysis. So as an alternative, I measured the time that it took for a flashlight that starts in the eyes, how long it takes to reach the brain. Because as I told you in the beginning, that speed of signal travel over the neuron is dependent on that level of myelin present. What I saw here was, indeed, when I stopped the action of the blue PD4D Pac-Mans now, this time was reduced. The speed was therefore increased. The signal reached the brain faster. And when I looked at the brain of these animals, indeed I could see that there was more myelin, which is then responsible for that increased speed. Now, in my study, I always think it's important that we can see whether our things that our findings mean something for a patient eventually. So I also made use of human-based experiments. But because it takes years and years for a compound to be tested in a patient, I investigated the direct role of my compounds on the cells of interest. So to make this a bit more clear, I isolated the immune cells that I wanted to alter directly from these patients, from humans. And this I did by using a blood sample. From that blood sample, I isolated immune cells. I activated them so that it mimicked a bit what was going on in MS. And then I treated them either not with my general inhibitor or my specific inhibitors. And then I evaluate how active are these cells, how good or how bad are they. What I could see was, just as like in my animal model, the general inhibitor, stopping all the Pac-Mans, decreased the level of activation of these immune cells. And something similar was happening when we stopped the purple Pac-Mans, the PD4B. This is in line with what we saw in these animals where less paralysis and better immune cells were present. Then finally, I also wanted to see whether the repair cells were positively influenced by my compounds, because I previously saw that the blue Pac-Man inhibitor was beneficial for giving more myelin in the brain. But we cannot just take a piece of brain of humans. So for this, I made use of urine-derived cells, which we reprogrammed until they were now brain cells. And then we evaluated whether or not they would make more myelin if we targeted all the Pac-Mans or only the blue Pac-Mans, the PD4D. By evaluating then the level of myelination, I could indeed see that, again, targeting all the Pac-Mans, the PD4 inhibitor was doing something good. And now the blue PD4D inhibitor, stopping the blue Pac-Mans, also increased the level of myelination. And this again is in line with what I saw in the repair mouse model, where also more myelin was present. So to summarize, my main findings were indeed that PD4, that general stopping of the Pac-Mans, was positive, but it had side effects. So therefore, in my PhD, I found that targeting the purple PD4B Pac-Mans is beneficial for tempering the immune system, while targeting the blue PD4D Pac-Mans was beneficial for giving or inducing repair. And this then supports the use of these selective inhibitors for treating MS. But what's very important is that inflammation and demyelination are not just happening in MS. It's also something very common for spinal cord injury, ischemic stroke, and peripheral diseases. Therefore, in Chapter 4, I also investigated the role of these specific inhibitors in spinal cord injury, in Chapter 5 in ischemic stroke, and in Chapter 6 and 7 on peripheral demyelinating disorders. And all these data supported the potential that these subtype-specific inhibitors have for treating multiple diseases. And with this, I'm at the end of my presentation. I want to thank you all for your attention, and I now give the word back to the prorector.
Thank you, Ms. Candidate, Ms. Schepers, and I must personally compliment you about the quality of your artwork. It's very well done, and it's really gives an extra dimension to this thesis defense. For the audience, we have today 7 members in the Corona, and there will be 3 online, which you will see later on the screens, which you don't see in the back, of course. And we have 4 members who are here present today. And without further ado, I would like to start, and I would like to start with giving the word to Professor Van den Hove. He was also the chairman of your assessment committee, and he's affiliated as professor in neuroepigenetics at our university.
Thank you, Mr. Prorector. Let me first of all, dear candidate, compliment you on this wonderful thesis, which does not just consist of a nice number of chapters that you shortly commented on today, but actually you did a lot of work next to what is described in these chapters, and I can only compliment you and also your promotion team for that. Also compliments on the presentation. You know I love Pac-Man, so far so good. But let me first ask you some further questions. I want to particularly focus on chapter 4, which is on spinal cord injury, where you touched upon in the very last slides. There you also did the same. You targeted both PDE4 isoforms, but also you looked at them independent from each other. Now there you conclude that the PDE4B, that's B from Ben, was not that effective, which then would have impact on the role of inflammation in that model used. So my question there is, what does this say about the role of inflammation in that model, and how does that fit with the general literature on the role of inflammation in spinal cord injury?
Highly esteemed opponents, thank you very much for your kind words, first of all, and for your question. So indeed, in the model that we use in Chapter 4, we didn't saw any beneficial effects of the PDE4B inhibitor. However, what's important to note is that the model that we used here was a hemisection model. So really, there was a cut in the spinal cord. You have some level of inflammation in there, but especially that hemisection model allows you to investigate the regenerative aspects of the treatments. Hence, if we would really want to investigate the immune system and the overactive or the suppression of that immune system with PDE4B, it would be a better option to use the compression model, where really this weight is dropped or the spinal cord is compressed. This leaves some sparing of your neurons, but there is a way more profound inflammatory reaction happening. So I do not want to exclude any potential role of PDE4B in treating spinal cord injury, but it's something that needs to be investigated with a more suitable model for that.
Yeah, makes sense. And yeah, I already understood from your answer that in such a model, you would not expect the same effects then potentially. Okay, then you know I'm always up for a good stats question, a short one this time. It's also in Chapter 4. So you mentioned that actually the functional recovery in the mouse model you look at using a two-way ANOVA with repeated measures. Mm-hmm. And I was wondering why two-way ANOVA, because well, what are maybe briefly your 2 variables looked at there?
So there was this Bazo-Mouse scale that we evaluated over time. So you have that repetitive measurement over time, and then the effect of the compounds of the treatment.
Ah, okay, because I was thinking that you actually mentioned one compound, the other, or the combination of the two being the 2 times 2 design, but you mean the repeated measures. Yeah. Then I think that's one way ANOVA with repeated measures, which is different, which might give you different stats result. But it was just a short one, and it has not been published yet, so you can still adapt theoretically. Then looking at that model, and that you already hinted at that also in your previous answer, the answer to the first question, we see actually in rodents that they have a different degree of functional recovery after spinal cord injury. And you can see that here, but you can also see that in other models of spinal cord injury. And I was wondering, is this species-specific effect that rodents are capable of maybe more also, more effective endogenous repair, is that related to all the things that you presented before? And is it actually related to then species-specific difference in PDE? So I'm not talking about the inhibitors now, but of course the isoforms. Maybe you read something about that? I was just...
Yeah, there's not that much done on the terms of spinal cord injury and the human situation correlating to that. There are, of course, some profound differences between murines and humans. Of course, I cannot deny that. But what I do think is, so what do I want to say? So what I do think is important is always in my chapter, I always wanted to validate my findings with a human situation as well. So indeed in rodents, there is much more capacity for regeneration and remyelination, we know that. But what I also saw in a later experiment was that human spheroids also were positively affected by the PD4D inhibitor, which also showed a beneficial effect here. So it may not be as strong as the murine model because there's a lot of this spontaneous recovery already happening, but I don't want to exclude that it's not doing anything in humans, maybe not to this extent, but if we can go from always in a wheelchair to moving hands, it would already be a way better option for the patients. So I do think it's more profound in the animal model, but it will definitely, I believe, work also or contribute to the care of patients eventually.
Okay, thank you very much. Yeah, I'm very happy with the answers. Considering the number of opponents, I give the word back to Correct.
Thank you.
Thank you very much. The opposition will be continued by Professor Huitinger. She's affiliated as professor in neuroimmunology, was a member of your assessment committee, and of course, on behalf of Maastricht University, I'd like to thank her that she took the train ride this morning all the way from Amsterdam to Maastricht. I'd like to give her the word.
Thank you, Rector. It's a pleasure to be here in Maastricht. I love this city. Dear candidate, I'd like to discuss your thesis with you, but not before I give you my compliments. It's very thick, heavy thesis. I learned a lot of it. I specifically liked chapter 3, as you may understand, given my background in MS research. Recently published, I think.
Yes.
Looking at OPCs, maturations, myelinations, different in vivo models, and looking at PE4D, B, and Ben, the P4 Ben is, well, in vitro, the myelination is most stimulated by 4D. 4B it's clear that it suppresses immune responses. If you look, and that does not work on myelination in vitro, but if you look in vivo, then it's clear that the EAE is suppressed by P4D, and no, vice versa. EAE suppressed by Ben, and the cuprisone is helped to remyelinate by 4D. So, looking at the clinics for multiple sclerosis, would you then choose to treat to block the P4B, the Ben?
Highly esteemed opponent, thank you very much for being here indeed, and for your question. If we translate this to patients, at the moment there's not this clear discrepancy between when is a patient RMS or when is a patient more in the progressive phase. It's still a bit vague in that intermediate phase. So I would, based on my data, opt for a first initial PD4B treatment if the person is in relapse remission to suppress the additional damage from being done. But if possible, but that needs to be figured out whether there is some compound interactions, but it would be ideal if we could treat patients early on with the PD4D inhibitor as well to enhance that endogenous remyelination as soon as possible.
Yeah, I must say that I was quite impressed by the effects of your PED4D late in chronic phase of EAE. It's quite, I did not see many publications showing such quite profound effects late in disease, likely due to remyelination, although you're not sure that is remyelination and that you see with the MBP expression.
So indeed, in the very late phase of the EAE model, we looked at the optic nerve to have some assessment of the remyelination capacity, but because it's so so atypical where all these lesions in the spinal cord are happening is hard to really pinpoint, especially when we want to see repair, where there was a lesion and which one is repaired now. So indeed, it's a hypothesis that it is due to the remyelination.
Yeah, still not sure. I was triggered by the PDs, looked up in the Barrett's RNA-seq atlas, looking which cells produce the D versus the B. And it appeared that the D actually is more expressed in neurons, more in astrocytes. Well, let me have a close look, as compared to oligodendrocytes. So I was quite surprised that you focus. It's clear there's remyelination, but what happens to the astrocytes and what happens to the nerve regeneration?
Yes, so in another study of a colleague of mine where I also participated in, we investigated the role of PD4D inhibition on neurons. And there we also saw that if we stop the action of PD4D, that more neurite sprouting or neurite outgrowth would be enhanced. So in vivo, it's not unlikely that there is this interplay between these neurons and the oligodendrocytes.
Yeah, it takes two to tango, doesn't it?
Yeah.
Given also time, I would really ask you these burning questions. What you don't know is that we, the Netherlands Brain Bank, are conducting a study in remyelinating and remyelinated lesions, look at gene expression with RNA-seq. And I looked up, of course, the regulation of the PDs before I came here, and I must say that I was, given the high effect you see in vitro and in vivo, no regulation, not in the remyelinated, not in remyelinating, and also in a non-remyelinating. So can you explain this, why it's not regulated, but adding does help?
Yeah, so for me, indeed, I believe that the PD4D is not altered due to the disease. I think that we can use the PD4D action as a mechanism to push the cells over this threshold of cyclic AMP signaling so that eventually this downstream cascade would be activated, that they will lead to differentiation. So I don't think the PD4D is a pathological marker of MS, but we can use it in our advantage because it's just an intrinsic mechanism of OPCs that they need that cyclic AMP signaling in the cells to bring them to the differentiation. Yeah,
looking at body language of the vertebrae still. Yeah, and EAE, the strong effects of the inhibition of the PD4B. Was that, and you give the blocker in the periphery, I assume, by the way, that it passes the blood-brain barrier, huh?
Yeah.
So, but it's in the periphery, so you didn't check the generation of the MOC-specific T cells in the periphery, so that is due to that aspect?
Yeah, so with our EAE model, we did evaluate the lymph nodes and the spleen for the same panel that we use in the central nervous system. There we didn't saw any effects. We didn't investigate the direct effect of PD4B on the lymphocytes directly. I can elaborate. So we didn't saw it in the periphery that it has an effect. And also within the bone marrow transplantation model, we did saw that there was this myeloid absence of PD4B, which was beneficial. So it's not unlikely that lymphocytes may also be altered by that PD4B inhibition. But what we saw or what a previously study also showed is that inhibiting PD4 does not directly alter T lymphocytes for its interleukin 17 reduction, but it does this via the monocyte. So there's this intermediate player in this cascade.
Yeah, now I see a sign. I thank the candidate for the questions and give the word back. Thank you.
Thank you very much. Then the opposition will be continue, but we now move online. So I would like to welcome Professor Bock. He is affiliated with the Department of Pharmacology and Toxicology at Maastricht University, and I would like to give him the word. Professor Bock.
Thank you. Now I see myself. Dear candidate, it was a great pleasure reading your thesis, and I also thoroughly enjoyed your talk. It was very clear, very well in time, very informative. I have a large question first that I will chop down into two parts. Given your strategy of inhibiting PD4B and 4D, you would still hit all of these 4B and 4D molecules in the entire body, suspecting that you would give these drugs to a patient or in optimized form. So the first chunk of the question would be, what side effects would you expect by inhibiting other PDEs, the same PDEs in other cell types? And the second part of the question, I would like to, how you would elaborate on more targeted approaches to bring your inhibitors into the cell types that actually matter for your disease.
Esteemed opponent, thank you very much for your kind words and your question. So indeed, the known side effects of PDE4 inhibition are these gastrointestinal problems. We are aware of that. So we evaluated those with the PDE4B and PDE4D inhibitor. However, on the test that we used, we could not see any indications of this emetic side effect, while we could see this with our PDE4 inhibitor. So we do not think, or so far we have no indications that it's causing this profound emetic side effects. However, indeed, PDE4B and PDE4D are also investigated in cardiac experiments as well, and their different effects of PDE4B and PDE4D inhibition are seen on arrhythmias and so on. But these are mainly in these deficient animal models that are deficient for the whole PDE4D or PDE4B early on. So it's hard to distinguish whether it's a direct PDE4D absence for a long time that is causing this effect, or rather lowering the action of PDE4D is also causing this. So far, given that we administered the compounds for 65 days to our animals, we had no clear indications that it caused these side effects, but it's something that needed to be or needs to be elaborated on in the future. And regarding the second part of your question, if we want to make this rather cell type specific, did I understood correctly? If we can modulate this more specific?
Yeah, more targeted approach.
Yeah, so for this, I hope to continue this research, because that's something that I also want to elaborate on, because I want to see what is happening downstream. Can we identify targets that are different between, for example, neurons and oligodendrocytes, which are both positively influenced by the PDE4D inhibitor? Is there this discrepancy which downstream signaling cascade is causing these different effects in these cells? If this really is divergent, we could use this as a more specific approach. We can use this downstream modeling. Also, there are new ways that they actually try to modulate, for example, the drug delivery to MS lesions, because MS lesions are highly they have a high level of fibronectin, for example, and there's also a lot of studies that try to make these capsules where they actually embed some compounds, and it will be released when there's a fibronectin high or rich environment. That could also help in aiding the drug delivery to be more targeted and more precise to avoid possible side effects.
Mm-hmm. Then the second question is, you said in the beginning that cyclic AMP is a good molecule. So having more of cyclic AMP would impose a good effect. I know that's a simplified view, and it's most likely true for immune cells, that it's anti-inflammatory. But given the fact that phosphodiesterase can localize into macromolecular signaling complexes, how would you revise the statement that always more cyclic AMP is always good?
So indeed, this was a rather simplified approach to envision that cyclic AMP or cyclic AMP can be a good molecule. However, it is really these nanodomains and these particular pools within a cell where cyclic AMP is elevated that cause the right downstream activation and is then causing the right or aimed for biological action. So it's not my approach, or I don't think it's good to increase cyclic AMP to the max in the whole cell, in the whole cytoplasm, but it would be better to modulate these nanodomains, these pools, with cyclic AMP levels where they need to be increased, and also do this in a controlled level. Maybe it's enough to just push the cell towards or over a threshold and not keep that cyclic AMP rise to be continuous throughout a very long period. So the more localized, the better, I would believe.
And following what you just said, targeting the catalytic center of PDEs may not be as precise as you want it to be. Could you give an example of a possible treatment strategy that would not involve a chemical molecule that targets the catalytic center of PDEs?
One of these advances that has been made are, for example, the PROTACs of the lab of Professor Bailey. There it actually uses a ubiquitous degradation of, for example, only the short isoforms, so that the long isoforms are kept. But a problem there is that it's not PD4B or PD4D specific. So it's PD4, all the same class of isoforms that are being targeted. So then it's not a D-specific molecule anymore, but it's rather an isoform class-specific molecule. But they already indicate the great advances that can be reached in order to make a compound more selective than the ones that are available right now.
Mm-hmm. Sounds good. I have no further questions.
Thank you very much, and exact on time. My compliments. We remain to be online, and I would like to give to Professor Nagels. He is affiliated as professor in neurology at the University Hospital in Brussels, and I'd like to give him the word. Professor Nagels.
Thank you, Professor. I would also like to start with congratulating Ms. Schepers on an impressive body of academic work, resulting in several papers and even patent applications. You showed that your work has potential applications in several clinical neurological domains. And for our conversation, I would like to focus on multiple sclerosis. Now, there you have shown interesting results, both on efficacy of this type of compound with regard to myelin, and also on tolerability with the emetic effects. And you also studied papers on clinical trials that have been done in the past with this class of compounds. And those trials were actually a bit disappointing. So if you combine all those insights that you have made in your PhD, what would your advice be for starting up a clinical trial with the compounds that you studied?
Opponent, thank you very much for your kind words, first of all, and also for your question. So indeed, there are a lot of clinical trials using a PDE4 inhibitor that have failed. For example, there's also the Ibudilas trial, which is a more recent trial, where Ibudilas is targeting different families of PDEs, but also MIF, for example. And that one actually made it until a clinical phase 3 trial, but now it's suspiciously quiet around it. So it had demonstrated to be efficacious in reducing the degradation of white matter, so it kept the brain volume on a good level, so it didn't decrease that much. But what's very important in that Ibudilas trial, what we learned from there, is increasing the dose, increase the effectiveness of Ibudilas. So increasing the dose was good, but increasing the dose was also accompanied with these side effects again, which were typical seen for that PD4 inhibition. So I believe that by targeting now these subtypes, we can, in a more safe manner, increase the dose to reach that higher efficacy level. Because I also believe that it's a positive thing that PDE inhibitors have made it to the clinical trials, because it has shown previously that it has the potential preclinical, but it's not perfect yet. It's not there yet. And I think by modulating and targeting these subtypes, we can make big advantages in that. But what needs to be done first is look at a more wide safety panel, because what we did here was evaluating the medic potential in rodents, but there's a big important difference in one amino acid in humans compared to rodents. So the target binding of the PD4D molecule or inhibitor to the molecule is not exactly the same. So we need to evaluate whether it's also safe in primates, for example, before we can proceed to a human situation. But for the setup, I would rather look at the Ibudilas trial, because it made it to the clinical phase 3 level, and it had shown some promising results. So I would use that as a guideline as well for further development of these specific inhibitors.
And when you look at that trial, for instance, does it give you some ideas on the ideal length of the trial? The reason that I'm asking this question is that you mentioned a trial in which there was even an increase in contrast-enhancing lesions. I haven't looked at the paper itself, but there I wonder, is this an effect in the beginning, or was this over a longer period of time? And also, a second reason for asking this question is that in your work, I think on page 85, you show effects on myelin. And there I wonder, do your experiments show a faster recovery of the myelin, or are you sort of confident that the end level of myelination will also be increased by your studies? So there the question is, what would be the shortest but still acceptable length according to your findings for a first human trial?
Yeah, so that's indeed a very good question. I'm not a clinician myself, so giving an exact time window on that question is rather difficult for me. But I do believe that the repair of the myelin will be done. So the lesions that have OPCs, there will be more myelination. But also when we looked at the downstream effects of that cyclic AMP signaling, it also modulates, for example, HCN2 channels. And modulating HCN2 signaling has demonstrated to increase myelin length and myelin thickness. So also myelin remodeling was then beneficially enhanced. It needs to be elucidated whether or not the compounds that we use here are also targeting the HCN signaling in that way. But I do believe that we can remyelinate better, like with that myelin remodeling, but also that we can boost these OPCs to start myelinating. So there needs to be, of course, a sufficient time window in where this allows and where we can functionally evaluate the recovery of these patients eventually. But to give an exact time on how long we should treat these animals in a clinical phase is for me rather difficult to put a number on there.
Very good. Thank you for your answers, Ms. Schepers, and also thank you for this beautiful addition to my academic library. Thank you, Prorector.
Thank you. Then the opposition will be continued and we go back on site here. I would like to welcome Professor Somers. She's affiliated with the Department of Molecular Biology at the University of Hasselt, and I'm pleased that she can join us today here live from Hasselt. I give you the word.
Thank you, Mr. Prorector. Dear candidates, dear promoters and co-promoters, I first would like to congratulate you with a very impressive thesis, I would say. My colleague already alluded to it. I think the thickness of your thesis is, to me, it's directly correlated to the amount of work. So I really would like to congratulate you on that. I have a question related to statement 4, and maybe I can invite one of your panelists, maybe Ms. Jan, to read statement 4, and then I will try to continue with my question.
The choice of using PDE4B or PDE4D inhibitors highly depends on the specific disease pathogenesis and careful consideration of the type and timing of PDEs for subtype inhibition is crucial.
Thank you for that. So I would like to ask you to elaborate on this statement, and then more specifically for spinal cord injury patients.
Yes, so highly esteemed opponent, thank you very much, of course, for your kind words in the beginning, and for your question regarding the statement. So indeed, I believe it's strongly depending on when we should treat. First in our murine models, when we should start treating, how long we could continue, and also which one should we target before. For example, PD4B in stroke, PD4B in spinal cord injury, it has different effects, which may depend also on the concentration and the inhibitor use, of course. But besides that, giving the timing, for example, we need to understand first the pathophysiology good in order to modulate the effects that we want to see. For example, in spinal cord injury, you have a first immune reaction, and in the beginning that can also be beneficial. So it's not necessarily to treat with an anti-inflammatory drug too soon, too early in these spinal cord injury patients, which also allows us to have a better window in which we can administer these compounds.
So what would be the ideal time frame to use your therapy in the, for instance, acute or more chronic patients?
Yeah, what would, in my opinion, would be good is to start within the 24 hours, but not so to have at least these 1 or 2 hours, because in these first early moments, you have a lot of neutrophil activation as well. And in chapter 5, I think, we also demonstrated that inhibiting PD4B decreased neutrophil activation, so we could prevent the secondary damage from spreading. But what's also important is that we start with that PD4D inhibition that is pro-regenerative. So it would be ideal, in my opinion, to reduce that inflammation in the initial phase to create this good environment by decreasing the activation of, for example, these monocytes, these neutrophils, and that we then can stimulate the repair processes of remyelination and neurite outgrowth with the PD4D inhibitor. So a timed experiment would be, in my opinion, the very good next step to investigate whether we can use these both in one trial.
Yeah, and of course the spinal cord injury population is a very heterogeneous population. We have very young persons, male persons, and the more older people. So my question is, yeah, you probably have a claim on the use of your inhibitors in spinal cord injury. Which patient population would you like to specifically target? The patients with a complete injury, incomplete, and do you expect differences in these patients?
Yeah, so indeed, a complete one is, I think, always hard to treat because it's a complete transaction. It really takes a lot before there is a functional recovery, and I think that's a very difficult population to target and to treat. Therefore, a partial spinal cord injury would, in my opinion, be the most promising population that we can treat with the inhibitors because you can enhance some signaling. You can use, if we inhibit PD4D, you can have this neuron signaling that's already enhancing the recovery of the other sparing neurons. But what I also think is that both rather young and more older people can benefit from this because a lot of these aging studies showed that, for example, metformin reduces or rejuvenates cells, and metformin and PDEs are acting synergistically. So I also think that with our inhibitors, we might have the potential to rejuvenate some cells, which might be, of course, beneficial for also a more elderly population. But of course, the young partial transaction patients would be the first fit for a clinical trial to look at some potency.
Thank you for that. Maybe a very small question at the end. Are you aware of some trials that are currently being run in spinal cord injury patients, and what targets are they targeting, and which patients, for instance, do they select?
Yeah, so I'm not that familiar with the spinal cord injury clinical trials. What I am aware of the studies that are going more into the clinical phase are a lot of these cell-based therapies, but especially regarding PDE inhibition in spinal cord injury, I don't think there's much done yet.
So I was at a meeting last week for the Wings for Life meeting, and I must say there are 2 clinical trials targeting no-go, and I must say that there is really room for improvement because the results are really not okay. So I think there's room for improvement. Thank you for your answers.
Thank you.
Give the word back to the prorector.
Thank you very much. The opposition will be now continued by Professor Brone. He is affiliated to the Department of Neurophysiology, and he considers himself as an engaged neuroscientist. I can see here in my voting. And he is affiliated with Hasselt University, and I'd like to welcome him, and I'd like to give him the word.
Thank you very much, Mr. Pro-Rector. Dear candidate, congratulations with your work, congratulations with your presentation, which was very well adapted to the broad public. We always like this very much. Let me ask you some questions, and I will go back to the molecular side of your study, which would not surprise you, I expect. You already mentioned this fibronectin and fibronectin inhibiting the OPC differentiation. So, how do OPCs sense fibronectin? Are there therapies based on this molecular mechanisms? And most importantly, might your PDE effect and cyclic AMP downstream effect be working via this fibronectin route or mechanism?
Highly esteemed opponent, thank you very much for that question. It's a quite challenging question, I believe. What I showed, or what I did show, was that the PD4D inhibitor was able to overcome the fibronectin-induced inhibition on the OPCs.
Exactly.
But what also a colleague, Professor Villa-Baron, showed was that this was, or that the GD1 1 alpha, I believe, is also capable of overcoming this via a pKa-mediated pathway. So I'm not sure about the real downstream signaling of fibronectin and whether it influences cyclic AMP directly, but it is a possibility.
And
I'm not sure.
So the fibronectin receptors,
Yeah.
integrin receptors, might they be influenced by cyclic AMP, downstream cyclic AMP pathways?
There are a lot of these gene ontology studies already done to look at these CREB binding domains. I don't know by heart whether there is a CREB binding domain in the fibronectin promoter causing more gene expression, but I believe that if it is causing this, we are still capable of overcoming this, because we saw with this continuous coding that our inhibitor is capable of overcoming this. And with those studies that I mentioned before, using that fibronectin to deliver a gene, I think that could also be in our benefit, that there is fibronectin and that we can overcome that inhibition as well.
Yeah, might be two flies in one. Okay. The next question would be, again, not a surprise, about figure 3.9, which is electrophysiology. And you very nicely show that there is a decrease of firing of these area postrema neurons that produce the emetic side effects, actually. Is this what you expected, a decrease, which is perfectly in line with what you want, lowering the frequency, but is this what you expected? I would have expected the other way around. Not a decrease, but an increase in the roflumilast.
Yeah, but there is this increase in roflumilast. It is significant based from the baseline firing rate. But indeed, we did not expect it, the decrease in PD4D inhibition to happen. And it's also not in line with the psilocin ketamine test, where we don't see a increase in anesthesia time. So it's not something that we expected. It's not statistical different from baseline. The GABR32A, because of the spread, and it is also a single neuron level. So we need to keep in mind what this does, of course, in a living situation. It would be perfect if it's even anti-emetic, but I don't think that will be the case. But we do see that increase in roflumilast, and we don't see it with the GABR32A, the PD4D inhibition. But that decrease in firing rates, we didn't initially expect it, no.
And again, short side question. What might be the mechanism here? So you had in your introduction, you described several downstream pathways of cyclic AMP. What might be your, at least, what do you think is my favorite?
Ooh, your favorite.
My favorite downstream mechanism of cyclic AMP in these cells?
HCN channel modulation.
Why?
Because they are the hyperpolarization channels that's regulate as well that's firing, right?
Exactly.
So there is this HCN potential. I also think here it depends. We use the pan-PD4D inhibitor, so it targets all the isoforms. And it would be interesting to see which isoforms are in the proximity of these HCN channels to cause that nanodomain signaling there. Maybe those we don't need to target if those are the ones different from the boosting mechanisms that would be ideal. But also the HCN channels can have beneficial effect, like HCN2 channel signaling can have beneficial effect on oligodendrocytes. So maybe we need to find a balance, whether it's HCN1 and 3, which are highly present in the area postrema neurons, the 2 not that much. Maybe we can alter these HCN channel activations separately.
Thank you very much.
Thank you.
There's room for one more question. There's room for one more question if you want.
No, it's fine.
Then we continue and we go back online. And I'd like to give the word to Dr. Fouquier. He's affiliated with the Department of Pharmacology and Toxicology at our university, and I'd like to give him the word.
Thank you, Mr. Prorector. Dear candidate, first of all, I'd like to address you my congratulations for your great presentation. and also for your very extensive thesis. And I'm, of course, extending my congratulations to your promotion team. So actually, my first question, I would like to get you a bit out of your MS comfort zone, and I would like to address chapter 5 on stroke. You've studied the impact of PDE4B inhibition in this model of stroke, and you've concluded that inhibition of PDE4B by reducing neuroinflammation reduced the ischemic lesion size. And of course, an ischemic stroke results from a decrease in blood flow, and the severity of the stroke is linked to the ability to restore the blood flow. So I would like to know, beyond the impact of PDE4 inhibition on neuroinflammation, I'm wondering if there is any possible effect of PDE4 inhibition on to restore cerebral blood flow.
Thank you very much for your kind words and for your question. So indeed, I looked into the things that are known regarding the PDE inhibition and blood flow. Of course, for PD5 inhibition and so on, there's a lot known. PD4 inhibition, it's quite sparse what is known. But what I did found was that in smooth muscle cells of the lung, for example, when there's ischemia and there's this upregulation of PD4B upon ischemia in these smooth muscle cells, and this upregulation of PD4B is then also leading to the PGC1 alpha upregulation, downregulation, and that is then hampering the mitochondrial stress. So what they also saw there was that inhibiting PD4B then was beneficial in these smooth muscle cells, and that it caused actually cells to be less motile, to have less of this vascular remodeling as well. So that was for me quite, maybe not surprising, but it was not something that we initially aimed for, because it would be ideal in stroke if we would have these collaterals that are formed to restore this ischemic stroke as well. But the fact that PGC1 alpha is then altered, it causes, so by inhibiting PD4B, we have that PGC1 alpha increase, which is then causing mitochondrial biogenesis, which could be good for preserving the functionality of neurons and everything of the other cell types as well.
And so, like, in the brain, do you know anything about the using maybe expression atlases of PDE4, like the expression of PDE4 in endothelial cells or VSMCs? You discussed the lung VSMCs, but like, do you know whether also in the brain this is the case as well? And that would be maybe a starting point.
Indeed, PDE4 and PDE4 isoforms are expressed in these endothelial cells. But what we saw previously was, for example, in the MS chapter, that there is especially an effect on the ICAM, if I'm not mistakenly, so the immune cell infiltration. But by doing these preliminary experiments with a tear or a real-time measurement of this leakiness of the endothelial cells, we did not saw an effect following inflammation and then our PDE inhibitor treatment. But then again, if we could preserve endothelial cell function under hypoxic conditions, which would then be more resembling to the ischemic stroke condition, we could maybe preserve more that function because we preserve these cells. But again, and a big note in this ischemic experiment is that it was a prophylactic treatment and not a treatment option. So we prevented damage, maybe because we preserved this mitochondrial function, but it was a preventive study, not a therapeutic one.
That's a good point that you are raising this limitation, of course, but I would like to actually jump to another question. In the general discussion of your thesis, you discussed the limitation that is inherent to the use of animal models, and you rightfully stated that the combination of models is key to validate findings. I really appreciate that you are showing these limitations. Actually, myself, not being very expert on iPSC derived cells, I've been asking myself questions about the limitations of these models as well, because it looks like magic. Also, what you have shown in your presentation today, you take cells from the urine and tada, they become OPCs, right? So like, can you a bit elaborate on the limitations of iPSC derived models?
Yeah, so indeed, iPSCs are of course not a perfect model. They are the next best thing that we have available, but it's not there yet. They do allow you to maybe more investigate the developmental aspects because you really reset them to a more stem cell-like phenotype, and then you look at that differentiation. So it might be more developmental than that's a really therapeutic one. Also because in my conditions I did not inflame them, for example, to mimic more that microenvironment. But they do have that advantage that they are human-derived. Because in my case, for example, with that PDEs, there is this difference in amino acids, which then makes the inhibitors different to act on these PDEs, and that's something that we cannot investigate in these murine models. But using those iPSCs, we now know that it at least is doing something also in this human context where that different amino acid is present. So they have some advantages, but also the big disadvantage is that they are not there in their environment from which they came from. It's more developmental. Also, the epigenetic signature might be different and so on. So there are some impairments, or it's not perfect, but it might be just the next best thing that we have available for now.
Yes. I really like your question. I was actually also aligned with what you said, and the impact of the environment, the cellular tissue microenvironment there that can also drive the cell differentiation, but also maybe regulate expression of PDEs. I was wondering whether anyone in the world, in the literature, have looked at expression of PDE, isoforms, subtypes in iPSC-derived cells in different conditions, hypoxia or does it differ a lot compared to, let's say, human samples or mouse samples?
So there are these, the real characterization of these PDE isoforms in the iPSCs is not done yet. That's also why I wanted to isolate these cells from the human brain.
Dear Ms. Candidate, you can finish your answer if you're not finished yet.
Yeah, so briefly, that's also why I use these laser captured cells, which then allows me to look at these isoforms in their intact-ish environment as well, besides the iPSCs.
Thank you very much. Ms. Schepers, Ms. Candidate, the time appointed for defending your thesis is now finally over. The degree committee will now withdraw to discuss not so much the quality of the thesis, this has been approved, but the way you have defended your thesis this afternoon. I request that you and your company await the results of all our deliberations and our return to this aula. Thank you.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. I know what's on the line. I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you won the battle once you've grown. South side, 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and not. Long road to the south side. 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and not. Years can't fly by now, don't waste all your time. 'Cause I'll go the extra mile.
Ga voor ons staan, je staat zo achter
Ja.
ons. Ja, waar moet ik staan?
Ms. Melissa Schepers. The degree committee here present has assessed the quality of your thesis and in particular the way you defended in an almost enthusiastic way your thesis this afternoon. In view of its positive verdict, but also taking into account your previous qualifications, the degree committee here has decided to grant you the degree of doctor. Professor van Mierlo is authorized to confer upon you this academic distinction in accordance with Dutch, and I also may say Belgium University customs. I invite your supervisor now to take the floor officially.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law, and in conformity with the decision of the committee here present, I hereby confer upon you, Melissa Schepers, the degree of doctor and grant you all the rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee and affixed with the official seal of the university.
If there's a photographer, it's now time to take a picture. If you want, you can move closer. Don't be afraid. Okay. I give the word next to Professor Hellings.
Okay, Melissa, on behalf of the URN School for Neuroscience, I will also hand you over the URN PhD Certificate of Excellence, a name of the URN board and his representatives.
And we continue with Professor Rutte.
Dear Dr. Schepers, on behalf of the Graduate School Mental Health and Neuroscience, I am also entitled to hand out this official certificate. The owner of this certificate has shown to have received adequate training in diverse academic skills and has gained relevant experience to become a successful and independent scholar. Congratulations.
And I give back the words now to the first promoter, Professor van Mierlo, who is going to speak about the laudatio.
Thank you, Pro-Rector. Dear Melissa, beste Melissa, ik ga het Nederlands doen. Beste dokter Schepers, gefeliciteerd met het behalen van je doctoraat. Je hebt het niet gestolen, je hebt het keihard verdiend, je hebt er keihard voor gewerkt, dus helemaal gefeliciteerd hiervoor. Ik denk dat werkethos, strive en toewijding termen zijn die verbleken naast jou. Je hebt dat de afgelopen jaren heel hard gewerkt. Sommige mensen dachten dat je effectief op biomed woonde. Dat is niet zo, denk ik toch. Goed, om jou de nodige lof toe te spreken, ga ik even terug in de tijd, een kleine trip down memory lane. Hiervoor ga ik 7,5 jaar terug. En ik denk de eerste keer dat we elkaar ontmoeten was in je eerste junior mastervak, waarin we een journal club deden rond macrofagen. En niet lang daarna vroeg dokter Mirjam Goufabriga of ik niet even een tutorsessie kon overnemen rond een groepswerk rond brain injury. Dat was in het vak van professor Somers rond preventie, screening, diagnose en therapie. En de bedoeling was dat jullie daar een groepswerk gingen maken rond brain injury. En ik herinner me nog heel goed, dat was op een vrijdagochtend, dat we eigenlijk voornamelijk gesproken hebben over hoe het was om in academia te werken. En de vragen van jou en van je collega's, ook collega's die nog steeds collega's zijn, gelijk Lise Evens en Paulien Baten waren daarbij, werd daar redelijk enthousiast rond gediscussieerd. En ik vond dat ook heel aanstekelijk. En het bleek ook bij jou aanstekelijk te zijn, want de volgende maandagochtend heb ik van jou een e-mail gekregen met de vraag of je niet een stage bij mij kan lopen. En we hebben dat ook vrij snel een gesprek daarover gehad. En niet lang daarna hadden we een overeenkomst dat jij bij mij stage zou lopen rond functionele modellen in remyelinisatie. En dat ging heel goed, we tekenden daar een mooie opzet rond uit, want we waren daar allebei heel enthousiast rond. En ik heb toen het voorstel gedaan, we kunnen misschien ook een stofje meenemen dat ik met Jo Mailleu nog getest heb, en dat ook wel iets bleek te doen op remyelinisatie, en dat ging over roflumilast. En we dachten, ja, weet je, we kunnen aantonen dat er deficits zijn, maar als we dat ook nog kunnen aantonen dat je die kan omdraaien, ja, dan is het helemaal leuk. En zo is je stage eigenlijk gestart. En aan het einde van die 10 weken durende stage had je een hele in vivo studie gedaan met postmortem erbij. En had je eigenlijk al je eerste paper al zo goed als klaar. Je was natuurlijk heel enthousiast om hierop door te gaan, maar een tweede stage op hetzelfde topic, dat gaat niet. Hè. Je moet je kennis verbreden. En daarom hebben we toen gezocht voor jou voor een interessante stageplek in een andere veld, een andere ziekte. En dat was een samenwerking ook met Rotterdam. Ben je onderzoek gaan doen naar de effecten van zeewier op cognitie en Alzheimer. En ook daar ben je dan heel enthousiast op gesprongen, maar je had één voorwaarde, je wilde wel betrokken blijven bij het PDE-project. Dat zou gelopen bij een andere seniorstudent. En dat heb je heel goed gedaan. Je hebt enerzijds heeft jouw seniorstage geleid tot 2 eerste auteurspublicaties en nog 2 co-publicaties. Je had daar ook een thesis over kunnen schrijven. En anderzijds was je heel nauw betrokken bij het project van Jassen. En Jassen was de seniorstudent op dat moment op dat project. En hij was heel blij met die steun, maar op een gegeven moment kwam hij naar mij toe, en maar om te illustreren hoe Melissa hierin stond, hij zegt, oh Tim, Melissa is een machine. En hij was dus danig onder de indruk van de capaciteiten van Melissa, en dat ben ik ook. Goed, dan op een gegeven moment zijn we dan beslist om voor een PhD-thesis te gaan, voor een beursaanvraag. En je werkt ook mee aan de TUL-aanvraag die we toen geschreven hebben, en waar we ook succesvol in waren. En dat is ook de reden waarom dat we als double degree hier nu staan. En je bent ook gestart als PhD-student op die grant en heel enthousiast gestart. Je had ook een senior-student die eerst ook op een PDE-project zou werken, maar dan later toch een beetje afdraaide. En daar zullen we later nog wel meer van horen. En ook hier groeide de interesse om bij je Paraninf om ook voor een PhD-plek te gaan. En op dat moment was er een call open voor een FWO-SB-grant, waar Essia voor is gegaan, maar waarvan jij zei, dat kan ik misschien ook doen, want als we het allebei halen, dan is er een extra kans dat er geld wordt vrijgespeeld om Essia alsnog te kunnen laten starten. En ik denk dat dat ook ondersteunt wie je bent. Hè. Je bent een teamplayer. Dus het was een heel altruïstische approach om dat op die manier aan te pakken, want je hebt je veel werk op de nek gehaald om dat te doen, maar het heeft je ook heel veel gebracht. En ik denk dat de uitkomst van die grant jou nog het meeste heeft gebracht. In eerste instantie, in december worden die beurzen aangekondigd wie dan de beurs krijgt. En bleek dat op dat moment Essia de grant had gehaald en dus een PhD kon starten. En voor Melissa kwam dat nieuws eigenlijk pas enkele weken later. En ik denk dat die 3 weken daartussen heel belangrijk zijn geweest in de ontwikkeling van Melissa. En ik herinner me nog een kerstfeest op Biomed in 2018, waar ik vroeg van, Melissa, is het oké? Nee, het is oké. Maar als ik al zie dat het niet oké is, dan is het heel duidelijk niet oké. En dat heeft even geduurd, maar ik denk dat je in de loop van de jaren daarna, toen je ook op die beurs bent gaan werken, dat je ook echt bent gaan groeien en het accepteren dat als dingen misschien niet lukken, dat je daar net beter van kan worden. En dat is een les die je nu voor je uitschuift en nog steeds toepast op alles wat je tegenkomt. Dus ik denk dat je die beurs heel veel geleerd heeft. Dus ik ben heel blij dat je in eerste instantie niet op de hoogte bent gebracht dat je ze had. Verder denk ik dat ook jouw technische expertise heel hoog staat. Je hebt ook de ruimte voor gekregen in die grant om daarop in te zetten. En ik denk dat waar de technische kennis van veel mensen stopt, die van Melissa soms begint, en daar worden zelfs taarten op verwet, dat Melissa het wel in orde krijgt. En dat blijkt dan vaak ook zo te zijn dat Melissa dat het toch technisch op punt krijgt om verder te kunnen met bepaalde projecten. Dus ook daar, ik denk dat je technische kennis, het is niet alleen een theoretische kennis die je hebt, je bent ook technisch heel, heel begaafd. Dan denk ik ook dat die beurs, die 4 jaar, je ook de ruimte hebben gelaten om te groeien. Hè, want je had dat Roflumilast-verhaal, je had ondertussen ook het PD4D-verhaal rond Schepers uitgewerkt. Je was aan het denken over een inflammatieverhaal rond PD4B. En je hebt me dan ook overtuigd, ik herinner me nog de tuinhuismeeting, om het zo te noemen, waar we de data of in de paper of in het tuinhuis zouden zetten, waar we dan beslist hebben om 1 grote paper te maken. En je hebt me dan ook overtuigd, en ik heb nooit iemand geweten die met zoveel enthousiasme heeft verkondigd dat ze 65 dagen lang 2 keer per dag dieren wil gaan behandelen. En Melissa heeft dat dus met heel veel enthousiasme gedaan. Dan denk ik dat je ook in die periode jezelf hebt ontwikkeld als onderzoeker, als lesgever, maar ook als entrepreneur. En cijfers zeggen niet alles. Maar ik wil toch even enkele cijfers weergeven over wie Melissa is. Melissa behaalde 5 travel grants om naar buitenlandse labo's, ook binnenlandse labo's te gaan. Zo is naar Rotterdam gegaan, naar Antwerpen, naar Leuven, naar Edinburgh, naar Luxemburg. Om allemaal onderzoekstechnieken aan te leren om ze dan in de home institutions terug op te zetten. Ze haalde ook 4 onderzoeksbeurzen, persoonlijke grants binnen. De Tulbeurs dan ook, dat is niet echt een persoonlijke grant, maar had je ook aan meegewerkt, je eigen SB-beurs, 2 Charcot-grants en ook een Stichting MS-onderzoek pilot grant samen met de groep van Via Baron. Verder behaalde je ook 4 prijzen voor oral presentations op internationale congressen, prestigieuze congressen, zoals de Gordon rond PDE's, waar je ook chair was van het seminar. Ook 2 posterprijzen. In totaal ondertussen al 23 papers gepubliceerd, waarvan 6 als eerste auteur en ondertussen ook eentje als laatste auteur, heel recent geaccepteerd. En nog 16 co-auteurpapers, die ondertussen al hebben geleid tot bijna 400 citaties. Dus ik denk dat het toch wel onderstreept dat Melissa heel uitzonderlijk is in het genereren van wetenschappelijke data en die ook te dissemineren. Ik heb al gezegd, je bent ook gegroeid als lesgever. Je kan heel begeesterend, met heel veel enthousiasme, mensen vertellen over waarmee je bezig bent op heel verschillende levels, van layman tot high-level professional. En ik denk dat het ook gereflecteerd wordt in je nominatie bij FHML samen met je paranimf Essia voor teaching, voor als beste tutor binnen FHML. Je staat ook steeds in de top 3 van het aantal gepresteerde onderwijsuren aan de U Hasselt. Je was genomineerd voor de wetenschapsbattle. Je hebt een opname van de Universiteit van Vlaanderen verzorgd. Je neemt deel aan wetenschapsdagen. Je helpt in het weekend op MS-run dagen. En misschien nog het meest indrukwekkende is dat je in je periode van je PhD 30 studenten hebt begeleid. En op zo'n manier dat meer dan de helft daarvan ook nu nog in het onderzoek actief is. Dus dat denk ik dat ze voor een groot stuk ook aan jouw enthousiasme en jouw aanstekelijk enthousiasme te wijten hebben. Je hebt ook leren valoriseren. Hè. Je bent betrokken bij een van onze patenten die toepassing vinden in jouw onderzoek. Je hebt ook contractonderzoek helpen uitvoeren, helpen organiseren. En het meest belangrijke denk ik is dat je doet niet wetenschap voor jezelf. Je doet wetenschap voor patiënten. Ik zou aan je andere paranimf kunnen vragen voor hoeveel patiënten? 4. Maar dat is natuurlijk meer dan 4. Melissa, naast onderzoeker, onderwijzer en entrepreneur ben je bovenal een prachtig mens. Hè? Je weet heel goed waarom je bent wie je bent en je weet ook van waar je komt en aan wie je dat ook te danken hebt. Hè? En dan kijk ik ook bijzonder naar je mama. Ik denk dat jullie heel veel kansen hebt gekregen, dat je mama die voor jou heeft gecreëerd, dat je ze ook hebt genomen. En ik denk dat je mama heel fier op jou mag zijn en ook andersom, dat je dochter heel fier mag zijn op de mama. Dus ook felicitaties aan de mama. De doctoraat is ook deel van de mama. Ook aan Kevin, de partner van Melissa, ook proficiat. Ik denk, dus Kevin Jansen voor de Belgen in de zaal, niet de acteur. voor de Nederlanders in de zaal, niet de procesregulator in Maastricht. Hij is veel beter dan beide. Ik denk dat hij in de afgelopen jaren zoveel presentaties van Melissa heeft mogen aanhoren dat we als Melissa uitvalt gewoon een stand in kunnen nemen, want Kevin kent dat allemaal. Goed, dan zou ik misschien nog willen afsluiten met een kleine anekdote op vraag van de co-promotor. Moest je volgende keer naar een congres gaan, pas op voor de vogels. Ik denk dat het verhaal is het volgende. We gingen naar de Euroglia-meeting in Porto en Melissa was zo enthousiast toen we terugkwamen van de eerste congresdag dat ze zich verschrok van een meeuw en op een trapje van 3,5 centimeter hoog haar voet trachtte te breken op 2 verschillende plekken. En die voet stond helemaal paraplu naar de verkeerde kant. En het eerste wat ze zei in tranen, ik zal morgen toch nog wel naar het congres kunnen gaan. Hè? Ja, dat is niet gelukt. We kregen ook te horen dat Melissa 6 weken thuis zou moeten blijven, maar ze heeft de dokter kunnen ompraten dat ze toch na 2 dagen terug op het werk kon verschijnen, omdat ze ook wel andere dingen deed dan in het lab werken. Dus Melissa, ik hoop dat ik nog heel lang met jou kan samenwerken. Het is heel aangenaam samenwerken met jou, je hebt een hele positieve energie. En ook namens het team wil ik je dus nog proficiat zeggen, proficiat Melinda. Ik wens je een prachtige carrière toe. Dan geef ik nu het woord terug aan de prorector.
Dear young doctor, I prefer to continue in English because we do have quite some English students and also staff here in this audience. On behalf of Maastricht University, I also like to congratulate you with your degree. This is basically the highest degree which you can earn here in the Netherlands. And also in Belgium, I think. Yes, but I have to be that. In my congratulation, I also like to bring my congratulations to your team. So the team who put you, who raised you and guided you over all these years. And I think we also should have a little special attention today to your first promoter, who today brought his first PhD students to his defense. And I think we should applaud for him as well. In my congratulations, I continue to your family, and you already mentioned this here, to your mother or your mama, like I heard now quite often, and your partner. I also like to thank the members of the Corona. We were with 7, and they all have some tough questions to you. I like to thank the members abroad online, both from Amsterdam, from Leipzig, from Brussels. And I also like to mention to the students who are sitting here in the audience, you have seen today a very nice thesis, a very nice presentation, and a very well defense. And the candidate has shown that she knows what she talks about. That's one thing. But what's also more important, that she loves to talk about it. And that's also what she presented today. And I hope to see in the near future many of you also here defending your thesis here as well. And by saying this, I'm going to close this officially defense. For those of you who are not familiar with that, I have a hammer for this over here. And I use my hammer and I close this defense. How are we going to continue? It's the following. There is a reception. Maybe even a glass of wine and some pastry. I'm not sure if it's there. We're going to make some pictures here, and first of all, we make the picture here with the online representative, and we make a second picture on the stair on the hallway of our university, and then after about 10 minutes later, we will join you also in the reception hall. And I hope